Good morning, everyone. I am Cyrus from NextGen Scientific. Can you hear me?
Yes.
Yes.
Thank you for attending this webinar sharing session, CRISPR Transfection Nucleofector Technology for Efficient Genome Editing. During the webinar, you may type your question in the chat box and it will be answered accordingly afterwards. For today's webinar, we are proud to have two honorable speakers. Firstly, I would like to welcome our first speaker, Dr. Noor Azian binti Muhammad Yusuf, a talented researcher from Parasitology Unit, Institute of Medical Research, National Institute of Health. Dr. Noor Azian is a researcher of parasitology at the Parasitology Unit, Infectious Disease Research Center, IMR. She graduated as Bachelor Honor in Biology from USM, Master in Science from UPM, and PhD in Structural and Molecular Biology from University College London. She was also a diploma holder in science from UITM and a postgrad diploma in applied parasitology and entomology from IMR. She served at the IMR for more than 25 years. Her research mainly focused on malaria parasites as well as some work on hermits and protozoa. Her research interest is in anti-malaria drug resistance, malaria recombinering, in vitro and in vivo cultures, applied parasitology as well as entomology, monoclonal antibodies, genetic population, and CRISPR-Cas9. She has served in various consultancy capacities to IMR and NIH. Among the more recent assignments were appointed as reviewer for the 22nd NIH Scientific Conference, subcommittee member for laboratory and diagnosis of malaria, panel member of the Research Evaluation Committee for NIH, associate editor for IMJR, expert panel committee in parasitology field for Encyclopedia for Infectious Disease in the Malay language with Dewan Bahasa and Pustaka, and other various activities in the area of expertise and training supervision. Currently, she is also a deputy dean for the APE school and course supervisor for a few master students. So let's welcome Dr. Azian for her sharing session.
Thank you, Cyrus. I just share my presentation here. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning and salam sejahtera. Thank you to the chairperson, NextGen, for inviting me to share my knowledge on malaria research with a special reference to Lonza Nucleofector. I will divide my presentation into two sections, which are, the first one is a brief introduction to malaria, my journey with Lonza Nucleofector during my malaria research work. So, without further ado, I would like to start this presentation by citing one quote. Behind the statistic in graph lie a great and needless tragedy. Malaria still takes the life of an African child every minute. This is my favorite quote, which first I came across in 2012. This quote was said by the WHO DG at that time, Dr. Margaret Chan. Although it's long ago, but it's still carrying the same message and the same goal, which are to lessen and eliminate malaria infection. What is malaria? Malaria is an ancient and life-threatening disease caused by Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae, and Plasmodium ovale. And they are transmitted to human by mosquito vector belonging to the genus Anopheles. They completed their life cycle in two hosts, which is a human cytogonic cycles and also mosquitoes sporogonic cycles. And what is zoonotic malaria? It has recently become evident that some simian malaria parasite that typically infects forest macaque monkey can be transmitted by Anopheles to cause malaria in humans in Southeast Asia, especially. They are frequently misdiagnosed with microscopically with human malaria parasites such as P. sp. malariae. And since 1960, a total of 7 species of monkey malaria have been reported as transmissible to men by mosquito bite. And they are Plasmodium cynomolgi, Plasmodium brazilianum, Plasmodium illicei, Plasmodium knowlesi, Plasmodium innoi, Plasmodium swetzi, and Plasmodium simium. And according to WHO World Malaria Report 2021, in the year 2020, 29 of the 85 countries that were malaria endemic accounted for about 96% of malaria cases and deaths globally. This pie chart was extracted from World Malaria Report 2021. The pie chart shows in the year 2020 an estimated of 2.1 million malaria cases was reported, increasing from 2 to 7 million in 2019, with most of this increase coming from countries in the WHO African region. Nigeria, the Democratic Republic of Congo, Uganda, Mozambique, Angola, and Burkina Faso accounted for 55% from all cases. And meanwhile, four countries accounted for just over half of all malaria deaths globally. The countries were Nigeria, the Democratic Republic of Congo, the United Republic of Tanzania, and Mozambique. And over the last 2 decades, malaria affected countries have achieved tremendous remarkable success, but the fight is not over. This map shows WHO Report 2020, where countries with indigenous cases in 2000 and their status by 2019. This green color shows countries with zero indigenous cases over at least the past 3 consecutive years, which has been considered to have eliminated malaria. In blue, China and El Salvador reported zero indigenous cases for the third consecutive years and has applied for WHO certification for elimination. And also, the Republic of Iran, Malaysia, and Timor-Leste reported zero indigenous cases for the second years. And this maps in WHO reports 2021 showed some countries have changed from zero cases of more than 3 consecutive years has been declared as certified malaria-free area. And what about us? Where are we? This is the latest report on malaria cases for the year 2000 to 2021. They showed a total case had increased from 2838 cases in 2020 to 3688 in 2021. Despite the increasing number of cases, we are lucky no indigenous cases were detected, and therefore we are on the right track for elimination certifications. However, at the same time, we observe a trend of an average of more than 3,000 cases of phenolcide per year. So now we come to under part two of my presentation. I would like to highlight two research that has been done as well as ongoing. Both type of research were carried out in different settings and different countries, but using the same type of Lonza Nucleofector. The first research is about malaria parasite invasion. I was introduced to Lonza Nucleofector during my PhD at the National Institute for Medical Research, and I am in London, now known as the Francis Crick Institute. My work at that time was to investigate the malaria parasite invasion to RBC, how it happened, and who responsible for the invasions. This photo was taken during that time, and this is Tony Holden Labs member, and this is me and Tony, my supervisor. So how does the malaria parasite invade RBC? This diagram shows basic understanding of malaria invasion to host cells. Attachment of merocyte to erythrocyte surface. The merocyte then reorientate to bring the apical end in proximity to the erythrocyte invasions. The secretions of apical organelles allow the formation of moving junctions and make the invasion irreversible. In the same process, this is the surface protein I shared. And lastly, the PV, if parasitophorus vacuole of PV, is formed from the erythrocyte, plasma lemma, and drop secretion. And once the moving junction reaches the posterior end of the parasite seal itself in the RBC. How does the malaria parasite invasion missionaries work? A general model of the main components shows that they involve a signaling cascade, including calcium and inositol triphosphate that initiate the gliding and evasions, a range of molecules for binding to host receptors. receptor and formation of the moving junction. The third one is actin and its binding partner that regulate polymerization, depolymerization, and filament connection to the moving junction. And the fourth one is the myosin motor complex that provide the force required, and that's where I work most of the time. So this is the diagram, the schematic diagram of ectomycin motor complex. Basically, myo A is the main component is myo A, where it's linked to the INC via a gladiosome associated protein of GAP45, GAP50, as well as a MTIX or myosin tail interactive protein. GAP M is a transmembrane protein which is highly conserved and specific to epithelial structure. Okay, the objective of this study is to investigate myo-A or myosin A and the interaction between other components of the invasion motor complex. And also, I would like to characterize of myo-A and myo-B and their cellular function. So this is to show you the schematic representation of the GFP tagging of myo-A by single crossover homologous recombination into the myo-A locker with primer for PCR and the southern blot probes and resection sites, the transfection process and the maintenance of the mutant parasite in vitro. So as you can see here, I use Lonza Nucleofector. The PF myo-AGFP parasite as viewed. This is after we successfully transfect the parasite. So we can see the PF myo-A GFP parasite as viewed by a live fluorescent microscopy. And it takes about 3 to 4 months to get an established Parasite myo-AGFP tag. And this is the diagnostic PCR on genomic DNA, just to show you that we got an integrated GFP myo-A into the parasite genome. So this is the schematic representation of the GFP tagging of myo-B by single crossover homologous recombination into the myo-B lockers with primers, and also I also show you here southern blot probes and root site. The PF myo-BGFP parasite also as viewed by the light fluorescent microscopy. This is already the successful story of transfection process. It's for myo-B, it takes about 3 to 6 months to get an established PF myo-BGFP parasite. And this diagnostic PCR to show you that we also have the integrated, also managed to integrate the F myo-B into the parasite genome. And this is to show you we not only do for P. falciparum, we also doing it in the P. plasmodium knowledge in vitro culture system. And the same results, as you can see here, we also successful in integrating the myo-A and myo-B in P. knowledge parasite genome. So this is just to show you the live microscopy imaging of PF myo-A. We follow 48-hour cycle of parasite life cycle in order to determine the time of myo-A protein expression. Myo-A protein was expressed at early trophozoite stage at 21 hours. Parasite development and the GFP signal were prominent, especially at the stage of early schizoid, and when the schizoid started to divide into merozoite around 40 to 48 hours of their life cycle. The role of myo-A as one of the main components for invasions of motor complex was strengthened with the evidence through GFP immunopreciptation. So as you can see here, although myo-A was expressed as early as 21 hours of life cycle, it formed a complex with other invasion motor complex protein around 36 hours of development, suggesting that it will be evolved during the invasions of RBC. Meanwhile, for myo-B, we found out that the express about at 40 hours of during the life cycle. Before that, it was not expressed. So using IFAT, we do some localization study of PF-myo-A and PF-myo-B with other motor protein complex. So PF-myo-A, as you can see, localized at the periphery of the parasite and co-localize with other gliosomal complex protein. Whereas for PF MyoB, it's localized at the apical pole of the parasite, and I will tell the the results later for MyoB. So this is the stepwise visualization of PF MyoA, which is you can see when free merozoite we can see the periphery of signal of GFP and then initial attachment, they when they invade and find the ceiling and also the stage. Whereas for MyoB, you can see the protein itself during this stepwise visualization. The MyoB did not move; it stays where it belongs, which is at the apical pole of the neurozoid. So this to show you that we want to see whether PF MyoA, again, this is the evidence to show you that PF MyoA was in fact together with the other main motor complex, which is MTIX, GAP45 and GAP50. And also we can see MyoB, it doesn't appear to be in favor with the other motor complex protein. So as a conclusion for this study, TF-MyoA, GFP forms a tetrametric complex in the gliadiosomal complex, and PF-MyoB localized to the extreme apical tips of neurozoid and not part of the gliadiosomal complex. So its real function is still under investigation. So now we come to research number 2, which is malaria research conducted in Parasite IMR, ranging from basic research to applied research such as malaria transmission mechanism, submicroscopic, in silico modeling and docking, female malaria, and R&D for development and improvement of diagnostic methods. Research 2 is one of my research which is ongoing related to anti-malaria drug resistance, and this is all my team members in this research. And that is where, again, Lonza came knocking to our door in Malaysia. So I start this with also one quote. Two things that are working really well against malaria are strong and safe drugs and insecticides. And the real concern is losing these two to resistance. This quote was by Profit White. Drugs have been used to treat and prevent malaria for centuries. For example, bark from cinchona tree, which contain an array of alkaloids with anti-malaria properties, appeared in Western therapeutics in 17th century. World War II led to the introductions of chloroquine, chloroguanine, or progranil, and eventually amodiaquine and pyrimethamine. The war in Vietnam brought mefloquine and halofentine. Although the use of anti-malaria drugs has a long history, the emergence of anti-malaria drug resistance is relatively a growing concern. Anti-malaria drugs resistance usually emerge not long after their deployment. For example, chloroquine resistant forms of resistance of Plasmodium falciparum malaria first appeared in Thailand in 1557 and about plus minus 10 to 15 years of introduction. The same phenomenon was observed in other classes of the drugs. And artemisinin or acid combination therapy or ACT was introduced in the late 1960s in an effort to prevent the development of resistance to artemisinin-based drugs. The WHO recommended that they be used only in combination with other anti-malaria drugs. The first widely available artemisinin combination therapy or ECT was Cotem, which combined an artemisinin derivative artemether with a long-lasting antibiotic lumefantrine. The combination drugs proved to be 97 effective in curing the most deadly form of particular malaria. Despite the product effectiveness, the WHO sought to restrict its use to the treatment of uncomplicated cases of particular malaria, fearing that wider use could contribute to resistance. However, in 2010, WHO reversed the policy ACT have subsequently become the first-line treatment for falciparum malaria in many countries. And unfortunately, the WHO fears that the generalized use of ACT would hasten the development of resistance appear to have been justified. What is artemisinin resistance? Artemisinin resistance phenotypically defined as delayed parasite clearance following treatments of artesunate monotrophy or MACT at day 3. The marker calf13, several countries, including neighboring countries of Malaysia, have detected presence of polymorphisms in calf13 genes associated to artemisinin resistance. And in Malaysia, artemisinin resistant or the distribution of mutation or identified marker is not well understood. Scenario in Malaysia. In 2013, deployment of ACT as first-line drugs. In 2016, delayed parasite clearance has been detected. And this graph was from bahagian kawalan penyakit, showed a delayed parasite clearance on day 3, showing that through drug response surveillance program from 2014 to 2020. From year 2014, we can see the case reduction of day 3 positive after treatment with ATP, and it's increasing with higher case in 2017. Although the cases was decreased up to 2020, but we need to bear in mind that the reduced potency of the artemisinin is prelude to ACT failure. So, we kickstart our research on anti-malaria drug resistance in 2016. However, at this point of time, we only use small sample size, which is an archived DNA sample for falciparum, and we it's just like a preliminary study. Among the isolate, we found two of the sample has been associated with delayed parasite clearance on day 3. We also observed mutation, which was also reported um, and also the rest were unique to our samples or isolate with three of them. The mutation keep appearing in more than three samples, but this data is still not enough. So in 2017, we expand our research for bigger samples and more scope and completed the study in the year end of 2020. The result shows that we have two mutations that have been validated and caused delayed and resistant to artemisinin. According to WHO list at that point, the red color. We also observed two of our mutation has been also reported by others and has been associated as candidate for artemisinin resistance. One mutation was observed in 5 of the samples, which is unique to our study. The rest we considered as new mutation, which unique to our country, since it was not reported as well. Upon these results, we continue our study in 2019 with the aim to study if our mutation contributed to artemisinin resistance, and where we choose several mutations of interest to be further validated. Um, this is a simple analogy. I would like to bring you to a simple analogy of conviction of mutation that either they cause artemisinin resistance or not. So this is the suspect that any mutation that occur in our isolates, and when we do this, we call as innocent unproven, which we detect delayed parasite clearance, where the mutation has been correlated with slow clearance in this clinical study. And this is the conveyed, which is the K13 mutation, has been correlated with reduced in vitro drug sensitivity using this speech assay, or reduced in vitro sensitivity resulting from the insertion of calcium in transfection study. And lastly is the verdict, yes or no, whether yes or no they involved in the artemisinin resistance. So what we do, we generate a transgenic parasite of artemisinin resistant gene. The general objective of this study is to study and characterize cell-specific protein function, and due to time limitation, I will only go briefly on the topics. We select three mutations, which is S600C, L598G, and N537I from our previous study. The selection criteria for S600C and L598G mutations is the mutation has not been reported somewhere else by other researchers, but is frequently found in our samples from previous studies. While N537I has been reported frequently in our clinical samples and studies by other researchers, but has yet to be confirmed to confer artemisinin resistance. We utilize the CRISPR-Cas9 system where we introduce our mutation. Our system comprises of three main components, which are the Cas9 protein, single guide RNA, and a repair template. The single guide RNA is designed to guide Cas9 protein to target our intended location. Cas9 protein will then lick the protein target. This will trigger homology directed repair system (ATR), which require a homology strand for their repair reference. Our repair template processes desired point mutation will be the reference for the repair mechanism. Our workflow start by designing two single guide RNA upstream and downstream of the mutation. It is incorporated into our CRISPR vector, a plasmid vector expressing Cas9 enzyme. We then design a repair template and ligate it into PUC57 plasmid for synthesis and amplification. Point mutation is introduced into the repair template by side-directed mutagenesis, and the plasmid is linearized before delivery into the falciparum nucleus. Now we have two CRISPR vectors containing single-guide RNA and Cas9 enzymes and a linearized repair plasmid. These components are transfected by electroporation using Lonza Nucleofector, done at one time at 0.3110 kilovolt or 590 microfarad with the resistance in ohm set to infinity. Upon successful delivery, expression vector will undergo transcription and translation for synthesis of Cas9 enzymes. The two translated CRISPR system will introduce two double-stranded break and on-targeted health-specific genes. This will trigger homology directed repair system with our design repair template as reference when repairing the damaged strand. And the result, a mutant variant calcium will be generated. So to date, we have generated 5 established mutant variants of the falciparum, and more mutants are under construction. I only will show 3 of them here. The first mutant is S600C. It took 12 days to detect growth in our transgenic culture line. As a verifying analysis, we sequence parasitic DNA and align with the wild type as reference sequence. This is the result of that translation of our mutant variant sequence. As you can see here, the top sequence is wild type reference sequence, and later in blue shows transgenic protein sequence have successfully changed from protein serine S to cysteine C. And these are the things here to show growth in day 12 when it is first detected. Purple and magenta stain are Vigilsa, and they are falciparum parasite. And until now, the mutant variant culture line has stabilized. The next mutation is L598G. We detected parasite growth after 27 days. On the right is the result of DNA sequencing of our mutant variant. The sequence in blue shows the translated sequence has successfully changed from protein leucine to glycine. These are the things here shows growth in day 31 when it was first detected versus today, and the mutant variant culture line has been stabilized. The last mutant variant is N537I. We detected parasite after 39 days. On the right is DNA sequence result of our mutant variant. As you can see here also, in blue, a transgenic sequence has been successfully changed from protein asparagine N to isoleucine I. And these are the pins here to show growth in day 39 when it was first detected by microscopy, and the mutant variant culture now has been stabilized. stabilized. As an implication of our study, we are able to achieve our objective with a time and cost-saving approach. A conventional transfection method may take up to 6 months. Maintaining culture life for a longer period is cost-consuming. It is also a development for fast-fitting genetic engineering methods and can be referenced for future fast-fitting studies. And as a future prospect, this allows for the validation of artemisinin resistance associated mutation, allowing us to look into the effects of this mutation introduction onto function self-testing and pre-falciparum survivability. This also allows surveillance of artemisinin treatments and provide insight of efficiency of the current treatment. As a conclusion, we constructed more of the mutant as well as new design where we will GFP tag our mutant parasite so that we can observe through live imaging. Using Lonza Nucleofector proves to be efficient in transfection studies regardless of how you construct your transfection system, either with single, double, crossover homologous recombination or triple Cas9. And the growth of the mutant using CRISPR-Cas9 system may vary, but so far it can be detected as early as day 12, but not more than 12 months. Whereas using more conventional, such as single crossover homologous recombination, it takes about 3 to 6 months for detection and establishment. And now we are in the middle of doing some research essay to more validate our mutations, whether they involve in the Artemisinin resistance or not. And by that, I end my presentation and thank you.
Thank you, Dr. Azian, for this amazing knowledge and experience sharing. We are really glad that Lonza Nucleofector can aid in your research. And we hope that Lonza Nucleofector can continue help you in your future research too. So for now, we would like to open for the Q&A session for Dr. Azian. Is there any questions? Dr. Azian, we have one question here. Have you tried any other transfection device before?
I not been able to try, but my colleague tried using, I think, a different a nucleofector. But no, I was introduced because at that time when I did a PhD, there was two types or two brands of nucleofector, and I choose Lonza because it's from the appearance it looks more nicer, more beautiful and futuristic. And that's my criteria at that point of time. But I think it's more easy compared because I also have the opportunity to look when my colleagues done using other nucleofector. And for me, I do not know, but I like Lonza most.
We have another question from Dr. Noor Azian.
Yeah.
Dr. Azian, is your editing involve a knockout or specific base substitution?
Specific base substitution.
Okay, so doctor, you are using the specific base substitution.
Yes.
And then we have another question from Sheikh Ahmad Kamal Sheikh from NH also. Doctor, which type of Lonza machine that doctor use for transfection, and do we have the machine in the institute? This I can answer on behalf.
Yes.
We have the Lonza Nucleofector 40 in IMR, and I can introduce to Mr. Sheikh in future. Is there any more questions? Maybe Yunn Wen got some questions for Dr. Azian?
No, very nice data. Very pretty, very beautiful pictures, and I'm glad that the technology is useful for you. So for Singapore, my experience is I also support at least 2 malaria group in Singapore, and they're also using from the old machine, the Nucleofector 2B, and using the new 4D Nucleofector system when we launch it. So the two differences is because the old one was still using the metal cuvette. So the metal cuvette will kill the cells quite easily. So usually the parasite will not survive so much. But when they move into 4D, I think the overall viability have improved significantly because the material of the cuvette. Because the 4D that you're using is the conductive polymer. I think that's helped to improve a lot in terms of the viability. Yeah. Very nice data.
Okay, thank you.
Okay, thank you, Dr. Azian and Yunn Wen. So now we will move to our second speaker. Our second speaker is Ms. Yunn Wen. Yunn Wen is currently the Senior Sales Manager in Lonza Bioscience Singapore. She is graduated from National University of Singapore in Life Sciences. Yunn Wen has more than 17 years of industrial experience in cell culture, therapeutic media, genome editing transfection, endotoxin and mycoplasma detection experience. She's also consulting on automated and digitalized GMP manufacturing processes. So without further delay, let's welcome Yunn Wen.
Can you see my full screen first?
Yes, Yunn Wen.
Okay, good. Yeah, so good morning. Thank you for NextGen for organizing this seminar, and thank you for inviting me to speak in this webinar regarding genome editing. So I'm not as good as Dr. Azian, but I'm trying my best to explain to you what we have and what we have assist most of the researchers in genome editing. So today, the topic of my presentation will be on CRISPR transfection using Nucleofector technology for efficient genome editing. But I will covering 3 areas for the next 30 minutes or shorter. So I'll give a brief on that introduction of genome editing tool, which is CRISPR, which Dr. Azian have briefly mentioned earlier. And the second thing is I will be talking about how to use Nucleofector technology for transfection of CRISPR and Cas9 system. And also will be providing some technical tips from a case study for you to improve your transfection efficiency or knock-in efficiency. So this is CRISPR-Cas9 in more detail. So before actually CRISPR being discovered, there are another 2 very popular genome editing tool. It's called a zinc finger or talents. But the CRISPR is discovered much later, which is a much simpler genome editing tool. Compared to zinc finger and talents, when you are using CRISPR, the DNA binding domain is the nucleus binding domain and the nucleus do not need to be fused. And the second thing is the DNA binding part is the RNA and it's not the protein. So both of these features make the CRISPR is easier to design from this 2 perspectives. So it's very, very popular among research group. So CRISPR stands for clustered regularly interspace short palindromic repeats. So it's actually originally from a bacterial immune system to defend against their invading viruses. So this system have been adapted, discovered, and used in eukaryotic cells. So the specificity is actually driven by the so-called guided RNA, the one in red, and it will bind to the complementary stage of 18 to 20 base pair in the targeted DNA and has some additional sequence motif that help to form a complex with the Cas9 nucleus, the one in greenish blue. So the Cas9, the successful binding of Cas9 and the genomic target sequence must also contain the correct PAM sequence, the one in the yellow. So the PAM is an NGG motif adjacent to the binding site. So this Cas9 nuclease marked here will unwind the double-stranded genomic DNA and enable the sliding along the guide RNA to find its complementary sequence. When if they found it, they will do a cutting of the DNA and create a double-stranded break. So let's take a look what happening in the process inside the nucleus. So when this CRISPR-associated nucleus cut the double-stranded DNA specific in the genome and create a double-stranded break, this event will induce a cellular DNA repair process. So there are actually 2 types of repair process that can occur. So depending whether you have the donor fragment or not. So without a homologous donor fragment on the left, so the broken end will then rejoin. And this is called a non-homologous end joining or NHEJ in short. So this is important for targeted mutagenesis or gene deletion. So the other one on the right hand side is if there are a genomic allele or the donor DNA present, so there will be an insertion or replacement of the gene can take place, and this is called a homology dependent repair or HDR. So this is good for targeted insertion or gene replacement. So actually, what happened inside the cells? So this schematic explains there are actually three different substrate or three different approach that you can use for CRISPR. Be it you can use the plasmid method, like what Dr. Azian have shown. She have used two different plasmids, and the other second approach is you use the RNA method. And the last one, which is getting more and more popular, is using the RNP method. So let me explain what are the three different approach here. So let's start from the left, which is the longest process. So the DNA plasmid have to be transferred into the nucleus. So after the transcription and export of the messenger and the guided RNA, the mRNA need to be translated first to form a complex before waiting for the NLS signal of the whole complex to be retransported back into the nucleus to do the genomic editing. So this is the longest process using the plasmid. So the second method, which is getting popular, is mRNA, because mRNA is slightly shorter process. So mRNA can be directly translated into the cytoplasm, and then they form the complex, and then being transported into the nucleus and do the genome editing. So this is slightly shorter compared to the plasmid. But the one getting more and more popular recently for the last 2-3 years is the RNP method. So you're actually able to transfer the RNP directly into the cells, and that help to reduce the time because the process can start almost immediately after transfection. The RNA complex is able to bring into the nucleus and start the genome editing straight away. Because this is a very short-term action, it also will be much easier to control the nucleus actions. So from what we explained for these three different approach, most of this is very commonly that you need to have a co-transfection method because you need to co-transfect more than just one substrate. Example, you can have two plasmid, you need to have RNA with the single guided RNA, you need to have RNP or with the donor DNA. So a lot of different substrate you need to bring into the cells. So other method being a virus or other electroporator might not be as good as helping you to bring into the cytoplasm and the nucleus directly. So the CRISPR will have a lot of tremendous impact on medicine and biomedical research, as what you have seen just now in treating malaria. We also believe that the CRISPR will able to help to treat the diseases at the cause of the disease at the molecular level. So it helped to use this CRISPR technologies using Nucleofector to actually create a lot of stable modification primary cells for using for in vitro assays or in vitro disease modeling. And the second thing is you can now do the gene repair and gene replacement on all those immune cell or induced pluripotent stem cells, and it's very, very popular to use CRISPR for CAR T or CAR NK now. So you need something technology that is proven and is robust and able to help you to bring this discovery to the patient and help to save life. So Nucleofector technology is a very efficient, is a non-viral transfection method. So this state-of-the-art electroporation is very versatile and very efficient. So we are very honored and very proud that we actually have more than 10,000 publications that actually using Nucleofector technologies, and many of them in a very high-ranking and high-impact journal. So this technology is also using in a leading lab for example, Yamanaka Group in Japan to do iPSC generation, and we also have Carpentier and Dogna Lab for them to use this technology for their CRISPR. So this is a very easy to use machine and technology, and I'll explain later why is that so easy. And it gives confidence for researchers to use it and apply this for their routine experiment. And as I explained, once you have able to establish the optimized conditions for your cell type, regardless of the substrate being the DNA being a plasmid RNA or RNP or a protein, it doesn't really matter. The substrate can bring into both cytoplasm and nucleus together. So we also provide unmatched scalability from very low cell number to very high cell number, like 1 billion of cells. So, in quick summary, so what are Nucleofector principle? So every cell type of interest, we have a cell type specific solution. And regardless of what type of substrate of interest, you are able to use the electrical pulses, and we bring this substrate of interest directly into the nucleus and at the cytoplasm at the same time. So their pores are generated in the cells membrane and also the nucleus membrane during the process, and it allows the substrate to enter the cells and the nucleus at the same time. So this is a very simple principle. So if you could check, there are a lot of data that we have proven that we are able to bring into nucleus. That's why we call this electroporation that we call a nucleofector, because it can bring your substrate directly into nucleus. So what are the components in this nucleofector technology that is very important to a successful experiment? The first thing is the nucleofector device. So although this nucleofector device is a electrical pulse method, but this device is able to generate a very gentle pulse. So that helps to transfect those very fragile cells like parasite, like red blood cells, like immune cells. Those are very fragile cells. And we also provide the specific nucleofector kit. So this is a kit that I explained just now. In the past, when Lonza came out with the nucleofector, it was using a metal or aluminum cuvette. But we realized that the metal cuvette actually killed many of the cells, because after transfection, the aluminum ion will be released to the solutions that kill the cells. Just like electrical shock. So we realized that it's very important of the material of the cuvette. So we changed the material into conductive polymer. It's a plastic, but the plastic is able to conduct the electricity. So without the metal ion being released to the solution, you're able to help to improve the viability significantly. And we also provide a very detailed optimized protocol, not just on the electrical pulses and everything. We also have in the protocol, we're guiding you from step to step how to do this, how to prepare yourself, how to do the centrifugation, and help you to give the best you could find to do the transfection, to give you a very high transfection efficiency. So this is a very quick 5 simple step that you can use with nucleofector system. So the first step is you harvest the cell of interest. If it's a suspension cell, you just collect the cells number needed. But if it's an adherent cell, you just need to trypsinize it and then just count the cells needed to move into the next step. So the next step is important to keep the solution and supplement mix, and then mix with the cells, DNA, your CRISPR, your RNP, and then you transfer into Lonza cuvette, either the 20 microliter strip or the 100 microliter cuvette. After that, you select the nucleofector program, insert the cuvette and press start. The whole transfection is second. Straight away, you get it done. So it's very important. All the preparation step is critical. So after transfection, you just rinse the cuvette with the pre-warm culture media and then transfer to your culture dishes. The expression can be detected as soon as 3 to 8 hours after nucleofection experiment. So it's very, very fast. So this is important when your cells are not dividing. Example, neuronal cells. If you want to transfect neuronal cells, you are able to use the nucleofector to help you to achieve that, because you don't need to wait for the cells to divide before you can bring the genomic substrate of interest into the nucleus. So this is a very simple 5 simple steps for nucleofector X unit. So Lonza, we have celebrated our nucleofector technology for 21 years this year. So last year, during our 20 years anniversary, we have launched a complete advanced platform for the nucleofector system with different modular units. So core unit is something that you must have, because that is a controller. But you have the flexibility to get different modular unit that's suitable for your application. So the most commonly, or what we call the generalist, most of the institutions, at least in Singapore, we have this 40 nucleofector system in the institution. So this is very flexible because it have two different cuvette material. So this is the 20 microliter strip. This is a 100 microliter cuvette. So this gives you a very good flexibility to do medium throughput and also bigger cell volume for 100 microliter cuvette. So this is most of the experiment, if they need to do proof of concept, they'll be doing here. But of course, if you have a lot of CRISPR, you want to do a CRISPR screening, this 16 well strip will able to give you medium throughput for your screening purposes. But once you really have a huge siRNA library, or you have a big CRISPR library, or you want to create a lot of cell type, generate a lot of stable cell line, then this is a very good screener modular system called 96 well units. So this is using a small cell number because it's only 20 microliter, but you can run 96 different experiments in just one plate within minutes. So that's helped customers to achieve the throughput they wanted. And we found that this is the latest trend that people are trying to create a lot of do a lot of CRISPR library screening using this type of technology platform. The next one will be the specialist, which is our Y unit. So this is very good for neuronal cells. So neuronal cells, you can culture your neuron cell on 24 well plate and use this specific electrode, the deepening electrode, to put on top to do the transfection straight away. So in this case, for Y unit, you do not need to trypsinize your cells. So for example, neuronal cells, fibroblasts, or endothelial cells is something that you can transfect the cells in adherent. So this LV, or large volume unit, is our industrializer. So it's able to transfect the cells in 1 mil up to 20 mil. So it gives you to able to transfect 1 to 2 billion cells within a one transfection experiment. So this is very, very important for cell and gene therapy. And we have customers using this platform for the clinical trial for CAR-T now. So this is to explain that for nucleofector technology, we provide scalability to customers. Even though that you're in the research lab and you use a smaller X unit, but you can actually transfer the condition that you have optimized in your research up to the industry and clinical when you need it in larger scale. So it's very good for condition transfer from research to manufacturing. So this is the publications that we have found. So we are very honored that a lot of researchers have using Lonza Nucleofector technology and CRISPR, and many of them have published. And in just 2021 itself, we have more than 1,800 publications that using nucleofection for the genome editing. So how to start if I want to do something CRISPR? It's very important to know what is the cells that you are dealing with. Are you working with immune cells? Are you working with parasites? Or are you working with endothelial cells or cell line? So you can, I will encourage all the participants to register yourself with Lonza Knowledge Center over the website by just using your email address. And once you're in, you're able to key in the cell number of interest and then find what you can find in our database. So in this case, I use CD34 as an example. So once you plug it in the cell type, they will tell you exactly what are the solution needed and what type of platform you can work with and what are the transfection efficiency and viability using a DNA PMAC GFP plasmid. So then you're able to retrieve all the optimized protocol we have for different cell type. We have actually more than hundreds of optimized protocol that customer can use it. So we have, this is a tested and proven technology, so you can find many reference in the Knowledge Center. So for the last section of today, I'll be using one of the case study from second and Roof paper on RNP to explain to you how they use nucleofections on RNP and achieve the very good high knockout efficiency. So Seki and Roof, they published their publications in Journal Experimental Mental. The topic of their paper are optimized RNP transfection for highly efficient CRISPR-Cas9 mediated gene knockout in primary T cells. So even without the T cells receptor stimulation, it results in almost complete loss of target gene expression at the population level with more than 90% knockout cells in single transfection. So their publications have a very advanced optimizations in gene knockout in primate human and mouse T cells. So this group, they have tested many different parameters, which is very important to be to have a very successful genome editing. Not just only selection of RNP or CRISPR complex, but the cell culture conditions is also can give you a different type of the result, and I will explain further later. And they also do a very important thing. They titrate the best ratio of different components for genome modifications of the cells. So the first thing is they try, because for primary cells of T cells and mouse T cells, we actually have 5 different solutions. Although Lonza recommended P3 and a certain selected program, but customer wanted to find the best one they can find with their own single guided RNA. So they try all the 5 different solutions and all 15 different programs of each solutions. And after that, they find what are the best result they can find, the knockout rate. And they select the solution and the program, and then they further optimize from this baseline. So they do the titration to find whether increase of the guided RNA and Cas9 would that actually help to improve the knockout. And they realized that from 3 microgram to 6 to 9, actually the result is not very significantly. But they found that the ratio like the 3 to 5 and 5 to 1 is actually help to increase double or triple of the knockout rate. So very important to try to do the titrations of the RNP amount needed for a successful high knockout. So the next one is they are trying to use the guided RNA, but they target different region and see what are the knockout efficiency. So the 3 regions that they target, you are able to see that it can be 4 or 5 times, even using the same guided RNA, but target different will give you a different knockout efficiency. So it's good to check around the genome, make map, and then try to target the region and try a few experiments on this. And the next one that they are trying to do is they use a multiple guided RNA but target the same gene. So that will also help to increase the knockout efficiency. So in this case, they have proven that using 2 to 3 guided RNA target the one region is actually help them to improve the knockout efficiency up to 85 to 98%. So that is very important. So they also see, is the Cas9 protein use is also critical for successful knockout? And indeed, they find the gene art platinum Cas9 and true Cas9 V2, and they found that the differences is very significantly 2 to 3 times differences. So this is also one of the conditions that you can, if you're working with CRISPR, that is something for your consideration. So in summary, this CRISPR-Cas9 mediated gene knockout published with this group, for mouse and human T cell, they have successfully come up with 90% knockout efficiency for multiple trials. And it also found that double knockout can be generated with a similar efficiency. So it's very important, as I explained in one of the slides, it's important to do a titration to find the best Cas9 and guided RNA ratio. So you can check on publication what will be the best ratio, or you can do your own titration on your end to see what will be the best for your cell type. And we do have a recommendation optimal RNP, but I still recommend customer to do the further optimization on their end. But remember, when you have a lot of different, if you're doing co-transfection with 3 or 4 different substrates, very important that this publication have shown is avoid too much dilution of nucleofection sample, because the nucleofection buffer itself is trying to pamper the cell, able to make the cell in single cell, and they are also very gentle to the cell to make sure that the cell membrane permeability is at their best. So if you have different substrate and you dilute too much of our recommendation, the result that you get might not be that optimal. So Lonza recommended that for foreign buffer or total of your substrate should not be more than 10%. So you can also consider complex directly with nucleofection solution. So these are some of the publication and clinical trial that we have. And Lonza, we have supply primary cells and culture media transfections, cocoon automation system, endotoxin testing, up to MODA or our manufacturing services. So we help customer in cell and gene in CAR-T. So if you need to find anything, any technology to help you from research to clinical, you need to find something which is efficient, robust, and versatile, scalable, and safety. So Lonza Nucleofector, we have the therapeutic version of solution and consumable for large volume LV units. So we are able to help customers to provide them with the GMP grade solutions and material, and we were able to provide documentation and support for customer moving into clinical trial. So, yep, with that, I thank you for your attention.
Thank you, Yun Wen. And we can see that Lonza has really put much effort in improving this nucleofector, which has helped more researcher and industry area. And we in Exchange really proud to be partner of Lonza in Malaysia. So we are open for the Q&A session now. We have the first question from Tengku Faris. Do you prefer nucleofection or lipofectamine for hard-to-transfect cells like stem cells? Based on your experience, which method yield greater efficiency, and which nucleofector is suitable to transfect iPS cells?
Should Dr. Azian or should I speak on that? The differences between nucleofections and lipofectamine is lipofectamine is using a lipid method. So you are not able to bring your substrate directly into nucleus. So if you need a very high transfection efficiency, that could be a bit difficult to achieve. That's why people are using, a lot of them using nucleofections for their CRISPR genome editing method. And of course, I will say that nucleofection will have a very high efficiency because you need some support than just a chemical method. So Lonza solution itself is a chemical method. So we use a combination of chemical method together with the physical method, which is electrical pulses, to create the best efficiency that you can get. So in this case study that I shared, that's why you're able to have knockout 85 to 98% for RNP. So that is very efficient. So if you can check all the publication, you can see they also compare viral and also nucleofector. And I can't comment that, but you can find the publication, or even in this paper, they have shared what are the differences, because viral is supposed to be very high in transfection efficiency, but they doesn't find as good for their RNP in CRISPR genome editing. So which nucleofector is suitable to transfect iPS cells? So for iPS cells, because they are very small and because iPS cells is very precious, most of the customers in Singapore that I will always advise them to use the X unit with the 16 well strip 20 microliter only. Because for 20 microliter, you only need like 200 to 400,000 cells per reactions. So that's what usually customer will use for iPS cells. And the second question from Sheikh Ahmad Kamal, and he's asking, can the 4D unit be used for a large-scale transfection? Depend on what large scale. So as I explained, the 4D nucleofector family have the X unit, which is our generalist. In the generalist, the large scale in this case is 100 microliter cuvette. So 100 microliter cuvette, in that case, we are able to transfect PBMC, for example, up to 10 millions already. But would that be enough, or you need billions of cells? For billions of cells, we recommend customers to work with the 4D core unit with the large volume LV modular unit, because for that one transfection, the highest you can go is 20 mil. So you are able to transfect at least 1 to 2 billion cells in one transfection. So I hope that answers your question.
Thank you for the suggestion and advice from Yun Wen. Is there any other questions from the audience? I guess we wait for another 1 more minute if anyone is still typing the questions.
My personal experience with the parasite is when I go to a MNC and they are working with a parasite and they were saying that all the electroporator that they try on the parasite doesn't survive. They got 0% of the parasite survive for their electroporations. So I joined Lonza more than 10 years now. One of the first years that I do was to transfect the parasite. And I bring the machine down there to do the transfection. And after 24 hours, we analyzed the parasite. And we were so happy. Although during that time, just the very first experiment, we see only 10% surviving parasite, and they are all green in color, and they are moving, and they are moving. So that is very happy when I see it. So I thought this is a very efficient technology. Although parasite is not what Lonza initial came out for. We came out for those very difficult to transfect primary T cells or eukaryotic cells, but we didn't realize that many of the researchers use it for parasite for their malarial research as well, and we are glad that the system worked for them. So you can actually using this nucleofector technology for bacteria or E. coli as well, although it's not our recommendation because we're still focusing on our mammalian cell. But if you need a system for both eukaryotic and prokaryotic cell, this is a very good system for you to have in your institution. Does that help, Cyrus?
Yes, Yunn Wen, is really a good suggestion and sharing from your side. Yeah, and Dr. Azian has said there, Ms. Tan, I definitely gained a lot of info and would like to expand my research. So this is really help. And yeah, another question from Shah Ahmad. I have problem using the old unit that the primary cells after transfection go quick slow. Will the conductive polymer help with the viability and proliferation of cells after transfection?
So do you mean the O unit is the nucleofector 2B? Because for nucleofector 2B, that was the first devices that the founder found out, and they actually used the normal aluminium cuvette. But as I explained, this cuvette is made of aluminium. There are metal inside. So like you take a metal and you touch the electrical point and you will get electrical shock. So people can die also together with the cells. That's why the viability is not very good. But during that time when when the founder founded it, right, it's still the best to work with primary T cells. Just that a lot of customers say it's very good, but I see a lot of floating cell on top because that is physical cell death and the viability is very bad. And if you're using the old machine, you have to stop the experiment as soon as possible because the longer that you expose the metal ion with the cells, the more cell death you'll see. So when Lonza realized the problem, we tested a lot of material in the cuvette, and we realized that the conductive polymer, there's no metal, is actually very good because you don't have the viability issue as much, and definitely not due to electroporation. It could be the DNA toxic to the cell, but it's no longer so much reason of the electroporation itself. So from my experience in Singapore is we have seen most of the viability, it can definitely help you to improve at least 20%. So it's the same for parasite because they are very sensitive. So they have limited cycle as well. If you do the transfection using a nucleofector 2B, the cycle of recovery is longer. So I work with a customer, they work with a very popular cell line called CHO cells, Chinese hamster ovary cells. For them, in the old nucleofector system, they have to select using antibiotic for selection. So for them, they actually use a very high dosage of antibiotic for selection using the old nucleofector machine because only half of the cells survive, for example. But when they move into 4D Nucleofector system, after we launch the system, they try. And because almost 90% of the cells survive, they have to re-establish using a lesser antibiotic to do the selections, and they find the stable clone. So if you can move into the new system, you can try, you can look for next gene for the demo, and you should able to observe in some of the viability have improved, and also the expansion will be faster. So for CHO cell, for example, in the past when they use the old Nucleofector system, they need 2 weeks to recover, to improve the cell continue to grow and up to the cell number that they required. So it take a longer time. But when they use 4D, because the cell membrane are protected, so they can actually just 1 week they have achieved they just continue culture for 1 week in the shake fast, and they get the cell number needed, because the cell don't need to recover that bad already compared to the old system. So I will strongly encourage, but again, depend what is your what do you want to get. If you just need a couple of cells with high efficiency, I think all unit is still work perfectly. In the publication, we have a lot of publications still using the old Nucleofector 2B. But if you need 1,000,000 cells in and you need to have 950 cells viable, so those you have to try the new machine to help you to achieve a good high cell number surviving and also high transfection efficiency. You need to make sure that the cells survive before you need to make sure that the cells can express the protein that you want it to be expressed.
Yes,
Okay.
Mr. Shah, I hope Yun Wen has answered your questions accordingly. And if you have any further questions, we shall make an appointment in NIH. I quite frequently visit NIH also. And yeah, if you want to visit the Nucleofector unit with Dr. Azian, yes, we can make an arrangement with Dr. Azian together, and we can understand more about it. And we hope that we can help you in your research also. So I think, is there any other things that Yun Wen or Dr. Azian would like to share?
For me, just thank you for inviting me to this session, and thank you, Miss Tan, as well, for a lot of info. And I will contact Cyrus for more collaborations type of discussion.
Sure. Let me know if you're interested with the Singapore team. I can do the coordination with the Singapore team as well for you.
Okay, thank you.
So once again, thank you everyone for joining this webinar session conducted by NextGen and our two honorable speakers, Dr. Azian and Ms. Tan Yunn Wen. And we hope to see you all again in our next webinar. Thank you so much for attending. And later on, we shall send the feedback form to all of you, and hopefully you can revert back to us. Thank you so much. Bye. Have a nice day.
Thank you.
Bye bye.